Okay, welcome back, everyone, to session 3. So it's my pleasure to introduce our third session, and we are now moving on to optimizing current treatments. Again, we've got 3 talks. And as Katrina mentioned before, with this topic, I think there should be also lots of opportunities. We might be able to think of additional collaborations we could be having within the center and with Monash. So that might be something good to keep in mind during the talks. And it's my pleasure to introduce our first speaker, Dr. Rika Pai Mangalore. And she is an infectious diseases physician at Alfred Health. And she's also undertaking a PhD on dose individualization of beta-lactam antibiotics using therapeutic drug monitoring.
Thanks, Connie. Before I begin, I'd like to acknowledge the traditional custodians of the lands and waters and pay my respects to the elders past, present, and emerging. And I'd like to extend those respects to any Indigenous people in the audience. So I'm going to talk about therapeutic drug monitoring of beta-lactams as a tool to optimize the dosing in hospitalized patients. And as Connie mentioned, I'm an ID physician at the Alfred and a PhD student at Monash. This is the brief outline of my talk. So just before we begin, I guess we just need to acknowledge the burden of sepsis on health loss worldwide. And in 2017, according to a report from The Lancet, there were about 48.9 million cases of sepsis worldwide with about 11 million deaths. And in Australia, we have 5,000 deaths estimated every year amongst 18,000 patients admitted to the ICU. So what does that mean? It has an impact on the length of stay in hospitals. There's a high in-hospital mortality, and there's a disproportionate burden on First Nations Australians. In 2020, there were 21,663 AMR infections in Australia, and those were the 5 species that they looked at. And 1,031 AMR-associated deaths with those 5 infecting organisms. And the health costs were 72 million Australian dollars. And if you had Pseudomonas aeruginosa that was resistant to ceftazidime, there was a highest mortality with that infection, and MRSA bloodstream infections had the most prolonged length of stay. So what's important is early recognition. We know that. Targeted treatment is essential, and with targeted treatment, I also mean prompt and very appropriate antibiotic therapy. So just before we go into a TDM talk, we have to talk about PK/PD. So pharmacokinetics is basically the way the drug travels through the body. And that's when you have a dose and you have basically the drug traveling concentration versus time. And then that drug goes into the tissues where it exerts its pharmacodynamic effect, which is either its antimicrobial or toxicological effect. This is what is important. This concentration time curve is what is important for beta-lactams. And the PK/PD index that we look for beta-lactams is the free time of the drug over the minimum inhibitory concentration of the organism. And depending on the beta-lactam class, if you're using whether it's a carbapenem or a penicillin or a cephalosporin, the time that the concentration needs to remain over the MIC for that dosing interval is different. So it's the concentration can remain above the MIC for 40% if it's a carbapenem, 50% if it's a penicillin, or 70% if it's a cephalosporin, and you'd expect that you'd get a therapeutic effect. But I would argue and put it out there to you that we should not aim for that. We should aim for 100% over MIC, which means that the concentration of the drug should remain over the MIC for the entire dosing interval. And I'm even going to say that we should try for 4 times over the MIC, so a really high concentration above the MIC of the pathogen. And why is that? Because currently we use standard dosing. So everyone in this room, if you're sick with sepsis, you'll get the same dose of an antibiotic, beta-lactams, particularly when you come into hospital. But that's not the same as exposure because we are all different and we all behave differently with sepsis, right? So when you have a fixed dose, you can't say that you'll get the same drug exposure at the site of infection. So what happens at the site of infection is really important. And because we are all different, we will handle the drug differently. So I think we really have to challenge the standardized, regulatory-approved, one-size beta-lactam doses. And I'll tell you why they may not be relevant anymore. We've just briefly touched on how different we are. So that's pharmacokinetic variability. When you're sick with sepsis or any infection, you're quite different from the drug dosing studies that were done in the community for our usual workhorse beta-lactams, as we like to call them, which are the usual penicillins like piperacillin, carbapenems like meropenem that we use in our septic patients. So in a septic patient, there is a difference in the way they behave in terms of their cardiac output. So they have a very high cardiac output, which increases the blood supply to various organs, and particularly to the kidneys. And that increases the clearance of beta-lactams because they're renally cleared. So you may not have enough drug in the system to actually exert its action. And then you have altered fluid balance and third spacing. And beta-lactams are hydrophilic, which means that they follow water. So if they're not in the space where they need to be and they've moved out into interstitial spaces where there may not be infection, then there's not enough drug to actually exert its action. Similarly, with renal or hepatic dysfunction, you can have changes in the volume of distribution and clearance, and you can have unpredictable plasma concentrations. You add an extra compartment to the body, for example, dialysis or renal replacement therapy or ECMO, which helps with extracorporeal oxygenation. And you have a whole new compartment with the increased volume of distribution and variable effects on clearance. So all these reasons separate a septic patient from a sort of healthy adult volunteer that drug dosing studies were conducted upon. And these are just illustrative studies, really. I just wanted to put them up there to show you that there are changes that happen. The first two studies on top are on augmented renal clearance, which is basically an increased renal clearance of beta-lactams that occur when someone is septic. And that is because of the hyperdynamic circulation. So these studies have demonstrated that you will not be able to achieve good plasma concentrations. And we say plasma concentrations because they're the easiest thing to measure in patients. And they are kind of a surrogate for the concentration at the site of infection. So the first two studies basically demonstrated that if you have augmented renal clearance, which many of our septic patients have, you do not have good therapeutic concentrations. The second study down is, or the second column there, row there is the protein binding. There's changes in protein binding, and there are many beta-lactams that we use for serious infections, particularly flucloxacillin for Staphylococcus aureus bacteremia, which is highly protein bound. And if you have a low albumin, the flucloxacillin exposure can be very variable. And as I mentioned earlier, there's renal replacement therapy, ECMO, and then changes in body size all affect our drug concentrations. So this is basically illustrating that the changes in pathophysiology are really significant, and your standard dose may not really be the way to treat a septic patient. Coming to AMR, I think, I was just trying to find an analogy to make it easier for most people. If you see the first cartoon or the first figure there, if you have, say, 2 in a billion mutants in a normal bacterial population, and you're able to treat the patient with a drug with a particular dose, you may be able to manage with just a dose if they have in combination with a healthy immune system, which can mop up the remaining infection. But if you add those changes to pathophysiology that I just mentioned, and if the patient has had previous antibiotic exposure, that changes the scenario completely. And the drug that you give may not be able to work as well, and the immune system is also a little bit compromised. So you may end up having an infection that's subtherapeutically treated. And I've just used an analogy there in the environment. Someone was talking about environmental impact of antimicrobials today. So just with subtherapeutic doses or subtherapeutic levels of antibacterials in the environment can cause increase in antibiotic resistant genes. And that is in the laboratory where they expose E. coli to a sub-MIC dosing of tetracycline and demonstrated resistance. So we had some local data that we conducted at the Alfred. We looked at a pharmacokinetic study in our septic patients, and basically just want to illustrate to you that, sorry, I don't know why that didn't show up. Anyway, there was a graph there which showed a wide variation in the concentrations of antibiotics. So we did demonstrate that we had a 140-fold variation in piperacillin concentrations and 200-fold variation in meropenem concentrations. So, and if we were to look at the targets for beta-lactams for their efficacy, and even if we were to look at 50% over MIC, we couldn't achieve it in our patients. right? So we had 30% of the patients who could not really achieve that conservative target. And if you were to look at even a higher concentration at half the dosing interval, about 80% did not achieve it. And similarly, if you went for the really the higher target of 100% of the concentration remaining over the MIC for that dosing interval, our patients weren't able to achieve that. 50% of our patients did not achieve a good target, and 90% did not achieve that more sort of aggressive target of 4 times over MIC that we should probably try for some of our patients, especially those with resistance. So how does this matter? Is it really that significant? There was a study in 2014 that was conducted by researchers from UQ. And this study was a point prevalence study. And it was a multi-center study, and they looked at 8 beta-lactams. And similar to us, they found that 21% and 40% did not achieve that sort of target of 50% over MIC and 100% over MIC. But what they also found that patients who had confirmed infections in ICU, 32% were less likely to have a positive clinical outcome if they didn't achieve that conservative target. But if they achieved a good target of 100% over MIC, then they were 56% they had 56% likelihood of a positive clinical outcome. And these are, again, some more illustrative studies in various infection syndromes that we encounter in the hospital of patients who have, who have aimed for higher targets and have had successful outcomes. The first one is a ventilator-associated pneumonia, and they saw a clinical success was higher, clinical cure was higher in patients who had 100% 4 times over MIC. The second study was in bacteremia. Similarly, they found that if you had 100% over MIC, that patients had better clinical outcomes. The third study was in febrile neutropenia. Similarly, if you had a higher FT over MIC, there was a better clinical response. And finally, again, a mixed study of different clinical syndromes. They found that clinical cure was better when you aim for 100% over MIC. And the other thing that pharmacokinetics helps is that we can establish toxicity thresholds. So the first study up there established, at least gave us an idea of toxicity thresholds for our common beta-lactams. And the second study demonstrated quite well that one of our beta-lactams that we use, which is Kefipime, has a concentration-related toxicity. So we know the toxic threshold of Kefipime now. And this has been demonstrated in several other studies. The problem is that it's difficult to demonstrate emergence of resistance in the clinical setting. And there are barely any studies that show optimizing doses with emergence of resistance. And there are some hollow fiber infection models that it should be intuitive that you know that if you haven't given someone the proper dose, then that there is a chance that they will have emergence of resistance. But that can't be clinically demonstrated in real time in our ICU patients. But the 2 studies at the bottom there, the hollow fiber infection model studies, have demonstrated that the higher the inoculum, the higher the chance of resistance. And there was a systematic review on emergence of resistance and dosing of beta-lactams. But most of the studies in there were animal model studies and preclinical studies. But they came up with a threshold for beta-lactam concentrations, which is a trough to MIC ratio of greater than 4, which equates to 4 times over MIC. So what are the risks of inadequate exposure? Poor clinical outcomes, persistence of infection, risk of toxicity, risk of emergence of resistance. So what is the optimal dosing regimen? What should we do? And you obviously know the answer because it was in my talk. It's therapeutic drug monitoring is one of the ways of actually optimizing doses of beta-lactam antibiotics. So in this, what we do is we measure the plasma concentrations, and then we target the dose to the plasma concentration and the infection syndrome. So you decide on a predefined target, and you aim for it, and you increase the dose or decrease the dose depending upon what the level is to achieve that desired concentration that you want, that you believe would be efficacious to the patient's infection syndrome. What is the evidence behind all this? Not great in terms of clinical studies. We did a systematic review, and we had 1,103 participants from 9 studies. There were variable studies from across the world, and they were predominantly treated with anti-pseudomonal beta-lactams. So just quickly, there was maybe a 15% reduction in mortality, but that did not reach statistical significance. But we did see an improvement in target attainment, of course, because you will be adjusting doses to the levels that you see. We did see an improvement in clinical cure and in microbiological cure. So, as I said, there were studies from across the world. They were quite heterogeneous, and all critically ill patients who may or may not have been septic were included. But I think we can take away that TDM-guided dosing does result in more precise, more individualized doses when you compare to the standard one-size-fits-all dosing that we use currently. So we do believe that there can be an improved clinical and microbiological cure. There is definitely better target attainment. And TDM can help with providing the maximum tolerated dose. For example, if you know the toxicity threshold and you have an organism that has a higher MIC or a high burden of infection, high inoculum, and you want to push the dose to an extent that you believe will be efficacious based on that infection syndrome, then you can actually do that with TDM, because you know you can quantify the plasma concentration and by surrogate the exposure at the site of infection. So it really does kind of take away the guesswork, the dosing in the dark sort of a situation in patients with high PK variability. And this is similar to what I just said. We believe that early target attainment would mean that if you get the right target and you achieve the right target within 48 hours of the infection or earlier, then you can hope for earlier sepsis resolution and reduction in the infection burden, and hopefully shorter length of stay and shorter duration of antibiotic therapy. So that's where I'm getting at. I think if you have a good preclinical study, and there are good preclinical studies that say that beta-lactams are efficacious at 40 to 70% over MIC, but then when you do it when you have clinical evidence, you know that they're okay at 50 to 100% over MIC. But if you're really trying to suppress resistance, then you have to push the dose to beyond that, to about 4 to 5 times, or maybe even 8 times over MIC, depending upon the infection syndrome. The limitations and challenges to TDM-guided dosing are that we don't have standardized dosing rules. We have assays that have to be developed in-house, and there are no commercially available or standardized assays. There are different methodologies. And sampling can be really cumbersome because patients may need 1 or 2 samplings per dosing interval and many times during their therapy. And the turnaround times, depending upon where you are, can be quite long. And serum concentrations may not always reflect the site of infection concentrations, because it's just a surrogate measure. You don't know how much is getting at the site of infection, and particularly with drugs which have a large volume of distribution. What's the future? And I think Connie's going to talk about it soon, but dosing software, model-informed precision dosing would be a good thing to have, good to validate and good to use in our clinical settings, which will improve how we do TDM. It'll be great to have some real-time assay development, point-of-care testing, I think, from morning's talks today. And the real-time feedback, for example, the biosensors that we heard about this morning, biomarkers, because infections are really difficult to get feedback on. If you have pain and you give paracetamol, you know that you're getting better. But when you have an infection and you've given an antibiotic, it takes you about 24 to 48 hours to actually know if the antibiotic is working well or not. And finally, I think what is sexy now is machine learning and artificial intelligence, and the combination of using model-informed precision dosing with individual laboratory parameters and individual clinical information that can be put into one system, which can then be linked with machine learning and AI to make it smarter. So each iteration will lead to a better dose in not just your current patient, but also your next patient that comes in. So that's my talk in a whirlwind. So I'll leave with this quote from Paul Ehrlich, which is hit fast, hit hard, and hit fast. I can't speak French, so I'm not even going to try. And I'll take some questions.
Thanks, Connie. Thanks, Rekha, for a great talk. I think this pertains to perhaps your actually last point about machine learning and AI and the parameters that you can feed into these this sort of system. One of the things that really also determines drug levels is obviously host genomics, and pharmacogenomics seems to get quite overlooked in our fields. The psychiatrists use it, and when you actually look at the outputs, there are extraordinary variabilities. We know that these cytochrome, these SNPs are really common. But if you look even just within, say, an SSRI class, that there are different profiles for drugs within an SSRI class. So it's actually very, very, very specific. And I think this is something that we should be considering in our patients. And while the turnaround times can be a couple of weeks, you could clearly produce them within a far shorter time and feed that into your AI model. Is that something that's considered?
It's a good point. With beta-lactams such as piperacillin and fluclox, they can be an allele that the organic anion transport allele in the kidneys may actually have an impact on the way the kidney handles flucloxacillin and piperacillin. And that can predispose patients to potentially renal toxicity. I think this is in really early stages. So I'm not sure how beneficial it is at this point for beta-lactams pharmacogenomics, but that's being looked at. In general, I think pharmacogenomics is an emerging field. I know it gets overlooked a lot. Yeah, I mean, certainly for our azoles, we do have that, but yeah, not so much for beta-lactams yet, but yes, it's being looked into. Yeah.
Thank you very much. So for this session, we were thinking we might try to keep most of the questions to the end to have the panel discussion. So for now, thank you very much, Rika, for this excellent overview, really, and raising all the important points. Yes. And it's my pleasure to introduce our second speaker, Professor Dina Liras, who is the Deputy Director of the Biomedicine Discovery Institute at Monash, as well as an ARC Laureate Fellow. And her laboratory focuses on developing immunotherapeutics and small molecules to prevent and treat infections by enteric pathogens. Thank you.
And I'll start with, you can hear me, can't you, at the back there? No? Now? Is it on? Is it on? Yeah, now it's on. So thank you to Ninad and to Anton for inviting me to speak today. It's been great, actually. I really like when different areas are brought together that we don't normally hear about. And so I always get a lot out of these sorts of sessions. So thank you for holding it. So what I wanted to tell you about today was, truly, my lab is a discovery lab. So to the clinical people, I'm going to tell you about some discovery research, mostly using animal models, but I hope it sort of translates into things that you can take away that will be useful for you. So we study gut infections. I'm going to tell you mostly about C. difficile, but we study other gut infections as well. And what we've tried to do, and what I'll present to you today, is to use that discovery research to develop non-antibiotic ways to treat these infections, to reduce the severity of these infections. I like, it'd be great to get some feedback on the things I talk about. So what I thought I'd start with today is to talk about the challenges of treating bacterial infections in the gut lumen. And so mostly we treat with antibiotics, and there are problems with that. So the problems start with we've heard a lot about microbiome today, and one of the biggest problems that we have is the off-target effects on the microbiome, so the reduction in the microbiome through antibiotic use. And that's really a central feature of C. difficile infections, so it's really important to the things I talk about. Microbiome obviously creates new disease susceptibility, but ongoing effects as well, because bringing the microbiome back can be very challenging. Absorption from the gut of antibiotics is a problem, because for bacterial infections in the gut that are not, intracellular infections that just stay in the lumen of the gut, you want the antibiotic to stay in that gut lumen. Absorption of that antibiotic is a problem, and so retention of the antibiotic in the lumen to treat that infection is a really important thing. Functionality of products delivered to the gut lumen is really important, because the gut lumen is a pretty hostile environment. So, we have pH issues, we have protease issues, we have lots of issues to deal with. So delivering products to the gut can be problematic in that they may not be able to function very well in that niche. And one of the other problems I think I'd like to raise, because I'm going to talk about it later, is that we think of gut infections as pretty much affecting the gut, but actually the extraintestinal effects are something that we've considered in our work, and I'll talk to you about that as well. So the reason that's important is it presents another place for treatment, which is not in that direct place of infection. So my lab studies gut bacteria. We're particularly focused on these opportunistic infections that arise from antibiotic use and microbiome disruption. We're also interested in mobile elements and antibiotic resistance genes. But again, we examine these non-antibiotic and antibiotics. So I said I'm going to talk about non-antibiotic, but actually at the end I'm going to tell you a little bit about repurposing antibiotics as well for enteric infections. So let's start with C. difficile. It's the side effect of antibiotic use. So microbiota disruption creates susceptibility, and C. difficile can come in and cause infections. It causes infectious diarrhea in hospitals. It's a problem in animals and in the environment. One of the biggest problems, which I'm telling you now because I'm going to touch on it at the end, is that it forms a different cell type called a spore, and that's critical to infection and also to recurrent infection. And critically for the work I'm going to talk about here is it causes disease by producing exotoxins. So they're toxins that are secreted out of the cell, go into the environment, and then they're the factor that causes the damage that we see. So the infection cycle is that C. difficile spores get into the gut. It's been disrupted by antibiotic use. The spores germinate. Vegetative C. difficile colonizes the anaerobic part of the gut. And then produces the toxins, which then go on to cause the disruption of the cells that we see in the epithelial surface. And this is the sort of thing that we see with C. difficile infection. Here's an uninfected bit of tissue from our mouse model, and here's an infected bit of tissue. So you can see that real disruption in that epithelial surface. You don't have to be an expert in gut to understand that this looks super duper different to this and doesn't look like it's going to function very well. So the toxins induce the damage. That's the starting point. We work with a bunch of industry partners. So all of the work that I'm going to talk to you about today has been with industry partners, and I'll point them out as we go along. So where I wanted to start was here, because I want to tell you about the sorts of studies that we've done in the lab looking beyond the epithelial layer. So on this side, we have a healthy person and a healthy gut. And here we have an intact colonic epithelium. We have stem cells here, which are responsible for repairing the gut upon damage. So here in the healthy person, they're intact and they're in the right place and they're doing what they should do. We have normal gut motility coming down here into the nerves. We have normal protease activity, and that's really important because I'm going to tell you later on that C. difficile disrupts protease activity, and that leads to exacerbation of disease symptoms. And we have normal organ function outside of the gut because the gut is intact and it's not permeabilized through toxin action. But here on this side, we have an infected colon, and the work that we've done really shows that we get a disrupted colonic epithelium. Here, first of all, we get that disrupted microbiome, and then we get C. diff colonizing. We get that disrupted epithelium. We get altered stem cell function. We get, I'm not going to talk to you about this today, but we get very disrupted gut motility with C. difficile infection. And gut motility really slows down upon infection, which is a bit sort of counterintuitive because it's a diarrheal disease. So I always thought a diarrheal disease probably means that the gut's moving faster. It's not. It really slows down. So we haven't published that yet. We get dysregulated protease activity, and I'll show you some evidence of that in a minute. And we also get, despite the fact that the infection is very much localized to the gut, we get this disruption of organs outside of the gut. We get this extraintestinal disease that is quite marked. So using all that information, what am I going to tell you about? I'm going to show you some evidence that with infection, upon infection, here on this side we have, and this is all done in our mouse model. So this is all tissue that comes from the mouse model. We really interrogated these integrity mechanisms that hold the gut together and allow it to function properly and make the gut not permeable. I think that's really important in gut infections, that permeability of the gut that results from damage here bacteria through toxins or other mechanisms permeabilizes the gut and lets things cross that gut barrier that shouldn't be crossing the gut barrier. And so, why is that an important thing? Because it really leads to worse disease and worse disease symptoms. So here, when we're looking at those microintegrity mechanisms of the gut that really hold the permeability in place, here's no infection, and here we're looking at factors that really sit between the cells. And here you can see this staining here is showing that upon infection, those mechanisms are very much disrupted, because you can see that staining between the cells looks very different. And here Ezrin is a marker that looks at polarity of the cells and how macromolecules in the cells are distributed through those cells upon infection. The Ezrin sits right on the top of the epithelial surface in a beautiful sort of green line. But you can see upon infection, that polarity of the cells is completely disrupted. So those microintegrity mechanisms are very disrupted. We published a study a few years ago now showing that the C. difficile toxins upon infection get right down deep into the crypts of the gut and actually damage and destroy the stem cells of the gut. So that's a really important thing, because it's the stem cells that repair the gut. And so if they're damaged, if they're gone, they can't do that repair work anymore. So we used among a lot of other experiments, I'm just showing you one snapshot here, we used an organoid culturing method to show that when we took stem cells from uninfected animals, we could culture these lovely little gut organoids out of those stem cells. But from the infected tissue, we couldn't grow any organoids, which told us there were no stem cells that were intact in that tissue that we looked at. Of course, there were some, but they were very depleted compared to uninfected. So stem cells are damaged. And when we looked at, and this is work that's just finishing up at the moment with my PhD student, Ash Rogers, we've shown that damage to the stem cells leads to a real reduction in the capacity of the gut to recover. And we've done a lot of sort of transcriptomics and proteomics to show what's going on during that recovery phase. And we've actually extended it out to 30 days post-infection. And even normally the gut recovers it turns over in 3 to 5 days. Here, what we've shown is that even at 30 days post-infection, the gut isn't completely recovered. And so that disruption of the stem cells and of the niche that looks after those stem cells means that the gut is not recovering. So even though the bacteria might be gone through antibiotic treatment, once that damage is done, that gut is not recovered in mice. I don't know how long it's going to be in humans. But in our mouse experiments, despite the fact that the bacteria are gone in a few days and there's no toxin left, that recovery of the gut is not complete at 30 days. So what does that mean? Why is that important? There's permeability of the gut that continues, which is a problem, which could contribute to disease recurrence, because you've got that capacity for the microbiome probably doesn't restore properly because it's not recovered properly. You've got permeability. There are lots of ongoing issues. So the first treatment that I want to talk to you about is one that we worked on with a company called Immuron, and they make a colostrum product. So colostrum, it's funny, I think about this colostrum product and people sort of don't take it seriously. They think it's sort of pseudoscience. But in fact, that's how we were treated when we talked about phages 15 years ago. And I remember putting in, small grant applications for bacteriophage 15, 20 years ago. And they were never, ever funded because nobody took it seriously. I think colostrum is a bit the same. So colostrum is a product that's made upon birth and is fed to babies, whatever that might be, baby humans, baby animals. Here it's baby cows. So what you can do with colostrum, though, is you can immunise your animals, and then in the colostrum there are antibodies specific to whatever antigen you've immunised those animals with. So we immunised cows with toxin B specific colostrum, so toxin specific colostrum with toxin specific antigen, I should say. So we made colostrum that had lots of antibodies in there that neutralised the toxins of C. diff. And what we did was we pretty much fed them to our animals upon infection or no infection or, a bunch of controls. And what we showed that was that here in primary disease, what you can see, I'll just point out the important ones. So here is no colostrum. So you can see these animals, this survival curve. So the animals all died within 2.5 days. And when we used a non-specific colostrum, so without the toxin antibodies in it, we also saw a similar rate of death of the animals. But when we used, you can see here this TCDB, HPC, the orange line, when we fed the animals with that colostrum, we rescued most of them from disease. And in fact, really rescued them from disease. They didn't show a lot of damage in the gut. They didn't get really sick at all. So we rescued them from really acute, severe disease, where normally the death rate is 100%. We also used that colostrum in recurrence experiments and showed that when there was no treatment, almost 90% of animals' disease recurred, whereas we really reduced that rate of recurrence with colostrum treatment. And I can't remember what that difference is, but it's about 60% or something like that. So colostrum directed at the toxins, which cause most of the damage, reduces disease severity and rescues animals from disease, and also reduces the rate of disease relapse, which was pretty amazing, actually, because this animal model is very, very severe. So delivering these antibodies to the gut lumen, they're not absorbed, rescues animals from disease. It neutralizes the toxins. It's a bit of a no-brainer. If you neutralize the toxins, you won't get damage. Why is this important for this sort of product? It's not an antibiotic product. It's really cheap to produce. It's sort of cents per dose. And you can direct it to whatever antigen you want to direct it to. So, and it spares the microbiome. So this is why I think we get this reduction in recurrence as well. So that's the first example I wanted to show you. Oh, and we're working with this sort of product on other pathogens, gut pathogens that colonize at high levels. So uropathogenic E. coli colonizes the gut to high levels, and that's probably the source for the bladder infections that come through. We've shown that we can make colostrum product that reduces, decolonizes the E. coli, and we made colostrum product against the surface, and we're talking about doing that now with a bunch of other gut pathogens that are important in hospitals, and we've talked to Anton about this as well. So I think this is a product that has a lot of use for gut infections, and hopefully we'll be able to explore that in future. So the second thing I wanted to tell you about was about a new facet of gut disease, which we didn't really know about before. So we did some work that showed that, I showed you that C. difficile infection damages the gut and really breaks that epithelial surface. As a consequence of that, what we found in this work here is that it's a wound, that, what happens in the gut with the toxins is a wound. So as with other wounds, plasmin is drawn to that wound, plasminogen, the inactive form, and then the active form plasmin is drawn to those wounds. So this is what happens in C. difficile infections as well. Here we have no plasminogen in an uninfected gut of, again, an animal model, but you can see how much plasminogen is drawn into the damaged gut upon infection. Why is that important? And I won't sort of point to everything because I'm going to run out of time. Why is that an important thing? It's an important thing for two reasons here, because plasmin is a protease, and a protease chews things up. And what we've shown in this work is that that plasmin that comes into the gut actually works against the host. It's not localized in the right place, it's spread through the gut, and it damages the gut further. And so, as well as the toxins doing damage, now you've got this protease that's entered the picture, and that's doing damage as well. So that's the first thing. But the second thing is that C. difficile takes advantage of that, and this plasmin coats the surface of spores, clips away the surface, and allows those spores to be activated much, much faster. And so they become vegetative cells and make toxins much faster than they normally would. That plasmin stays stuck to the spores, and the spores that are secreted out of the host are coated in plasmin. Does that help them? We don't know. There are experiments that remain to be done. But I can tell you that when we've taken spores out of humans, they're coated in human plasmin as well. So the spores that come out of humans are coated in a human factor, which may aid them in germinating faster, in evading the immune response, in functioning in a different way. So knowing that, we did an experiment where we used antiplasmin antibodies and nothing else. We didn't treat the animals with antibiotics. We just treated them with these two plasmin antibodies. And this was work that was done with James Wistock's lab. They created the antibodies. And what we showed was that, here we have, again, our untreated animals died within 24 hours. Whereas when we treated with this GO5 antibody, which inhibits the generation of active plasmin, we rescued most of those animals from severe disease. So that was really surprising. I didn't expect it to work that well, because I thought, oh, you've got to give an antibiotic as well. But in fact, treating this factor, neutralizing this protease, rescued the animals from disease. So here again is a treatment that is not an antibiotic treatment, that's treating a facet of disease that we really didn't understand before this work was done, and yet it rescues from disease. So the last example I want to tell you about is about extraintestinal damage that occurs from that leaky gut that you get from the toxin-mediated damage. So I showed you that earlier. You get a very leaky gut, stuff gets out of the gut, and we showed in our work that actually has a huge effect on disease severity. So the more the gut is leaky, the more disease severity you have. So here in this work, we showed that we get damage to the kidneys, damage to the thymus. I won't go into those details, but what I'll show you is when we give those animals a treatment that is completely outside the gut. So we injected these animals with, we worked with Merck on this and use a product, a monoclonal antibody called bezilotoxumab, and that's an antitoxin B antibody, so one of the toxins of C. diff. And what we did was we just injected those animals with intraperitoneally with Bezlo. We didn't give them anything else. So it was just with bezilotoxumab. And what we showed that when we treated with that antibody, we increased survival of those animals by almost 40%. We looked at those animals in detail. The gut damage was huge. It was still really high, but we managed to rescue a lot of those animals and reduce disease severity simply because we were neutralizing what was outside of the gut. So that treatment did not affect what was happening in the gut. It affected what was happening outside the gut, and it reduced disease severity. So again, here's a treatment that is not affecting the bugs, it's not an antibiotic, but it's reducing disease severity. And the last thing I want to finish on is that we also, I told you I wasn't going to tell you about antibiotics, but I'm telling you about antibiotics. So we've also done work looking at repurposing of antibiotics. And here in this work, in a 30-second, grab of this work, one of the biggest problems that we have is spores. So spores are present and in really, really high numbers in C. diff patients, 10 to the 6, 10 to the 7 spores per gram of faeces. The spores are really a problem for disease transmission within the hospital, within animal farms, within whatever. So the spores are the key to transmission, but the spores also play a huge role in disease recurrence. So if you could reduce the amount of spores that are made in the host, perhaps you could reduce both of those things. We never had a target. So in this work, we identified a target and an antibiotic that it completely inhibits C. difficile sporulation and reduces spore shedding. And here we showed that it completely prevented disease recurrence in our animal model. And so again, here's a target that we didn't know about. Here, can we make a product that can inhibit that target but doesn't act as a general antibiotic that will destroy the microbiome? So of course there are problems with this because it will still destroy the microbiome, but here is a new target and a new way of preventing disease recurrence, and so we're chasing that at the moment. I talked a bit fast, sorry. So these are all our collaborators, and I'm really happy to answer any. So before I finish, I'll just say, I think a lot of these mechanisms are not just C. diff specific mechanisms. I think when we start looking further, they'll be more general to enteric infections. And so the solutions we come up for C. diff, I think will also apply to other enteric infections. And I'll finish there. Thanks, everyone.
very much, Dina, for the fascinating presentation. Do you want to take like one question now, or any burning question now for Dina? Yep.
Thanks. It was a great talk on the new therapeutic strategies on C. diff infection. I was thinking about fecal microbiota transplantation, FMT, in treating C. diff. It seems to be really good at treating C. diff as well. Is it something that you could maybe use in conjunction with your new strategies as well? What is the field's general thinking or opinion towards FMT?
Yeah, I think FMT is pretty amazing, actually, and it's, microbes are really amazing. I think it's probably core to a lot of the work that will be done in the future for C. diff and modulation of microbiome and other things. I don't think we're quite there yet. I mean, it works really well, but it's a little bit sort of random, and people are reluctant to use it. But I think it will be developed. I'm really curious, the sorts of studies that we're doing now are asking questions like, that slowed down recovery that we see with C. diff infection, can we expedite that recovery by doing an FMT transfer? Can those microbes signal to the cells in the gut to tell them repair faster, do things faster, work better, make your cells join up better, and don't let the gut be permeable? They're the sorts of questions that I think we need to ask, so we understand the mechanism behind how an FMT works better.
So thank you.
Shall I introduce you? So I'll change hats now. So our third speaker in this session is Cornelia. So Cornelia is an associate professor at the Monash Institute of Pharmaceutical Sciences, and she leads a research group that integrates dynamic in vitro infection experiments with mechanism-based mathematical modelling to optimise dosing of antibiotics, and that's what you're talking to us about today. There you go.
Thank you so much, Dina. Very kind. So yeah, it's my pleasure to have the last talk here. And Rika has made a very nice introduction and touched on a lot of the points, so I can follow on. And basically, just from a high level view, infection outcome depends on relationships between the patient, the antibiotic, and the bacteria. And there are many patient characteristics affecting these relationships, as Rika has already mentioned. There are also drug characteristics, such as dosing regimen, mode of administration. And there are bacterial characteristics like susceptibility, resistance mechanisms, or biofilm formation. And also, we've got traditionally dosage regimen selection based on the PK/PD indices. As Rika has explained, we've got time above MIC for beta-lactams, and for other drugs like fluoroquinolones, we've got unbound AUC over MIC, so that's just the area under the curve of the antibiotic divided by the MIC. These have been really helpful and commonly used over decades. However, there are limitations that are recognized increasingly. And just as an example, we've studied 7 isogenic strains of Pseudomonas aeruginosa with different resistance mechanisms to meropenem, the carbapenem, and ciprofloxacin A fluoroquinolone. And if we expose these 7 strains to 2 times the MIC of meropenem, we still see quite a range of bacterial outcomes. Also, for ciprofloxacin, a different antibiotic class, if we achieve the traditional PK/PD target for all of the strains based on their individual MIC, we see this huge range of bacterial outcome in an in vitro study over 72 hours. So that's one example where the concentration response relationship for antibiotics is a bit more complex than the traditional indices. And there are other limitations that have been pointed out, such as we can't really incorporate the full time course of antibiotic concentrations and bacterial counts, or predict very well what's happening with heteroresistant strains. Also, they can't really be used for combination therapy or for integrating other data. However, we've now got other approaches which we can use to address these limitations, and that's what I'm focusing on today. There's been a recent position paper in Lancet Microbe, including a number of authors from Monash and from the center. And they'd also pointed out these limitations of the traditional methods and the key future directions for research, such as optimizing combinations, incorporating mechanistic insights into models, and then personalize the dosing. And because evaluating a lot of regimens in clinical studies to demonstrate efficacy is pretty challenging, we can maximize what we can learn from preclinical studies and mathematical approaches. And one very commonly used preclinical model is the so-called hollow fiber infection model, where we've got bacteria and we can expose them to the antibiotic concentration time profiles that they would be exposed in an infection in a patient. Now we can use this method to optimize dosing regimens for patients, such as critically ill patients with augmented renal clearance. And that's a problem because patients with augmented renal clearance basically eliminate drugs faster, such as meropenem, that's eliminated through the kidney. So we can see if we've got normal renal function, then this patient would have the meropenem concentration in black. But someone with an augmented renal clearance would have a much shorter half-life and concentrations decrease much quicker, which means there is a high risk of subtherapeutic concentrations and resistance, and we need to optimize the regimens. And that's just one example of how we can use preclinical models and mathematical models as an approach. So we can have such a isolate, a bacterial isolate from a patient in our preclinical model. And this example was an isolate that was actually quite susceptible to meropenem and susceptible to ciprofloxacin, but also heteroresistant to meropenem. So when we use our in vitro model to simulate the pharmacokinetics of ICU patients with ARC, we see here bacterial counts, we see a control in red, and ciprofloxacin monotherapy in purple, which provides initial bacterial killing, but as we can see, regrowth. Also, if we look at meropenem 1 gram every 8 hours in green and 2 gram every 8 hours in orange, this doesn't have a very good effect in monotherapy. Although this would have achieved the traditional time above MIC targets for killing. The combination, however, suppressed regrowth. And our modeling approach was able to describe all of these profiles. Now, importantly, as we've heard before, we want to know whether there's emergence of resistance during treatment. And we can look at that easily in vitro, where we see that meropenem as monotherapy in the orange and green had a huge amplification of resistance compared to the control treatment, and that's on log scale. And then the same happens with ciprofloxacin monotherapy, that very much increases the emergence of resistance compared to the control. The combination, however, suppresses resistance. So as mentioned, we can't really describe this with the traditional method, but the mathematical model was able to describe all of these curves and the observed less susceptible subpopulations simultaneously for all of the treatments and control. So we can address these limitations also for a heteroresistant strain. And then we can combine this model from the in vitro study with data we've got from patients. So we can use population pharmacokinetic models from the literature for patients, where we can predict what are the antibiotic concentration time profiles, then combine it with our model from the in vitro study, and predict what's the likely bacterial outcome in patients. And in this example, we can predict, let's say we have a patient group with an augmented renal clearance with a creatinine clearance of 200 mL per minute. And then we would get this range of concentrations for meropenem and this range of concentration time profiles for ciprofloxacin in the patients. So when we then put that into the mechanism-based model, this would then predict that for a combination treatment, such as 2 gram meropenem every 8 hours plus the ciprofloxacin, at least 90% of the patients would have suppression of the bacteria. And we can predict this also for different renal functions, such as even higher renal function. Maybe only 70% of the patients would be predicted to not have bacterial regrowth. And then if we are able to take a blood sample from the patient, so TDM, combine it with TDM, then we could use the information from the individual patient and basically predict what might be most likely. And this is just an example in terms of the ARC and the model. It has been used to inform patient therapy in hospital with success in patients with ARC. Now, concentrations at the infection site are interesting because oftentimes the infection is not in blood. And here we can also use mathematical methods in order to predict what might be the concentrations in tissues, such as in this case, we had cortical bone and we had patients with orthopedic surgeries and they received ciprofloxacin. So based on having data from patients and making a model that includes physiological values, we can predict these concentrations in different tissues, also in cancellous bone. And then again, this could be combined again with a model for the concentration response and pharmacodynamics to predict outcomes. Other options for these approaches with the preclinical models, we can look at, for example, metabolomics data. And this is an example using ceftolozane tazobactam against a clinical hypermutable Pseudomonas isolate, where we looked at simply 2 different monotherapies of ceftolozane tazobactam in orange and blue in the total bacterial population. Again, we saw there is an increase in resistance compared to the control in black. And then there were extracellular metabolites that tracked with the bacteria. So here, an example, ornithine could be modeled together with the total bacterial population. And very interestingly, some metabolites like ribose 5-phosphate actually tracked with the resistant bacterial population only. And we can do this in vitro and model it. Of course, the interesting future thought would be, can we use these approaches to maybe identify some bacterial specific markers for tracking biomarkers? And then lastly, of course, what we are interested in is, can we combine an analysis looking at all of the different factors? And that's a proof of concept study, which was a large collaboration with University of North Carolina through Monash UNC Pharma Alliance, where we had bacterial isolates from patients who received either ceftazidime, avibactam, or colistin, or the combination, and they had carbapenem-resistant Klebsiella pneumonia bloodstream infections. And here we were able to combine preclinical studies, mathematical modeling, and looking at genetic and clinical factors in one analysis. And interestingly, here are the 5 overall most predictive variables of 30-day mortality in the study. 3 of them were antibiotic resistance genes. 1 of them was the concentration of ceftazidime required for half maximal bacterial killing from the preclinical study and model. And from the clinical factors, hypotension was predictive. So to end, it's clearly a big challenge to apply these methods to really have the truly personalized treatment for patients at the bedside. However, there are opportunities to link with rapid diagnostics and things we've heard in session 1, and then include mechanism-based modeling, AI methods like Rika mentioned, to define optimal exposure targets and the dosage regimens we need in order to preserve the activity of current antibiotics and maybe inform how new antibiotics should be used. Thank you. Okay, so we have time for the panel discussion. If you want to, Rika and Dina, come here.
So my question to Dina. So you said that your findings can be translated into other infections. So I know that Vibrio cholerae, one of the gram-negative bacteria that causes similar infections like that, it causes diarrhea. Do you have any plan to see that's your findings is translated into that space or not?
So we have, I guess, where our starting point is the models that we have already established. So we have models for, I don't know, we've got, infection models for Enterococcus, for E. coli, for parasites. We've just published a couple of papers on parasitic infections of the gut. So my starting point would be our already established models. They're interesting because they cause disease in different ways. And so we're really curious to see if there are common mechanisms involved. So, for a pathogen that's an intracellular pathogen, what happens to the stem cells there? What happens to those mechanisms there? So I think that's where we'd start from. But, we can adapt our infection models, I think, to most pathogens except for the ones that won't infect mice. Yeah.
I have a question from Sarah McGuinness, who's zooming in for you, Dina. So can you make any comment about the regulation around production and advertising of the therapeutic bovine colostrum products? So she's saying in travel medicine, we often get asked about Travelan, an OTC bovine colostrum product for travelers' diarrhea. My understanding is that the claims made by the company, example, one claim is 90% protection, are based on CHIM studies. Chip, I don't know what that is. With, oh my goodness, ETEC specifically. Sarah, I wish you were here. And therefore not translatable to other pathogens. How did I go?
No, I understand the question. That's the important thing. So that product, so there is a product on the market called Travelan, and I don't know if any of you have taken it when you've travelled overseas. I wasn't involved in any of those studies, so I can't comment on the efficacy studies that were done. But that product is directed against intratoxigenic E. coli, and it's against surface factors and the toxin of intratoxigenic E. coli. So my understanding is that it works really well against that E. coli, but you have to understand this product. Colostrum is a general product that will have antibacterial effects, protective effects, soothing effects on the gut. And that's what it's designed to do. The difference between sort of normal colostrum and the colostrum that's in Travelan, which is an ETEC colostrum, and the colostrum we use, which is a toxin-directed colostrum, is that we have directed the response to particular antigens. And so, we know it works really well against our infection. Our product works really well against our infection. Would it work against really diverse strains of C. difficile? Maybe, maybe not. I don't know. And so that Travelan product, I can't comment on the data that was generated to show that its efficacy, but I know that it was directed against a particular enterotoxigenic E. coli and against particular surface factors, which we know can vary, and the toxin that's produced there. I don't know that answers the question.
You presented fabulous work on better dosing and strategies for better dosing with beta-lactam treatments in particular, and you did mention that one of the unmet needs at the moment is to have ways of measuring serum levels of the drugs in order that you can adjust the dosing appropriately. That made me think that Simon Corrie stood up in the first session and said that he wants to bring back tried and true diagnostics for point of care, low cost measurements of things in blood and serum. And so I'm wondering, have you guys talked?
A lot, actually. So we do have assays that are available. It's just that the assays have to be developed in-house. So there's different methodologies for the assays. It's cumbersome to have your laboratory to go through the whole process of developing it. So that's what I meant. But yeah, we've talked a lot, Connie and I. I mean, the ideal would be that we won't have to do that many blood samples and run that many assays. And if we have a really good model-informed precision dosing methodology available to us, then we could spare the patient of that many tests, as well as the lab of that many samples to run. So the burden on the infrastructure will be different. So the ideal world would be a really good MIPD software available to us with all the new things that are coming, for example, machine learning. But we need a lot of data for that. And your models have to be validated in your population. So every ICU or every hospital will have to have a model that's validated, isn't it? That's yeah. Do you have a comment on that?
Yeah, no, that's a good point. Or you could inform the model. Yeah, because the way these models work is basically you keep making them better. So you make the model and then you apply it and then you learn where is the model good and where do we need more data. And so it's basically this cycle of learn and confirm and improve it. Yeah. Simon?
Thanks, Trevor. So I was just wondering, just trying to learn this or learn a bit more about this, but with the TDM, I'm just wondering, you're mentioning measuring levels in serum. There is a great technology that's been licensed to a company, Neutromix in Melbourne, and it's coming. I think it's pretty cool. It's basically real-time seconds resolved drug monitoring, with a catheter in your arm or something like that. So I'm assuming that in a few years' time or something, that technology will be available to be able to do time-resolved monitoring of a whole bunch of antibiotics, because that's what they've been developed for in mice and rats so far. But if that's the case, then is the real challenge still being able to work out how serum levels track with other sites? Is that still the major challenge, or if that technology was around, would that make a big difference?
So I think the main challenge with having a biosensor-based sort of constant monitoring is extrapolating what you find. So from what I understand, the technology means that it will be in the interstitial fluid. It'll be measuring concentrations in the interstitial fluid. Now that again brings up the challenge of what is the concentration at the site of infection or in the bloodstream. So you need to make a lot of correlations, and the biggest challenge is to get the optimum dose. So what are we going to give the patient based on those numbers? Yes, I think if you have interstitial level that corresponds to a good dose, and then you know what to do with that level. What do I give my patient next? So if you know that, and you know that level that you're measuring in the interstitium is associated with efficacy, toxicity, suppression of resistance, whichever outcome you want to look at, then that's fine. But I think the challenge is to get that, to understand what to do with that real-time feedback.
And that's where you need to know not only the concentration of the antibiotic, but it depends on all of those other factors. So that's why you basically need to bring in this information from like diagnostics, like we've talked, and these things Nenad has talked with the new sequencing methods maybe, to work out. So one thing is know what's the concentration. The other thing is knowing what's the concentration profile over time, because it's the shape of the profile makes a difference. So how does the shape of the profile need to look for the infecting strain, for example, for the characteristics of the infecting strain? And then, if we know those two things, then we can sort of calculate back what should be the dosing regimen. We can do that. And for other sites, like other tissues in the body, there are these models where we can use physiological knowledge, and we know what's the blood flow and what's the tissue volumes and all of it, and the partition coefficients. So we can make some predictions, which is not perfect, but there are softwares around, and we can definitely do that as well.
Do you think we'll get to a point where you can use, we have those glucose monitoring things that people put on their arms, and then they use their phone and they scan. Do you think we could get to that, to measure?
That's a challenge.
That's what Neutromics.
Exactly.
Oh, is that what they're doing?
Yeah.
Right.
Microneedles.
And you can keep measuring the levels. Yeah, that's cool.
But haven't quite got the patch done. So I think Simon that I think that would be so exciting to have real time, because then we would get what Connie's explaining, the trajectory over time, and then our dosing. For beta lactams, we want time above MIC. You could all imagine a bell going off as the concentration drops below an MIC of your causative bacteria. I think it would be really exciting, but we're waiting for them to have developed something more than just these electrodes and wires, and we're very keen to do first in human studies with them, and I think that'd be really exciting. Yep.
Just a comment too, that the person who developed the initial technology is now on sabbatical here. So
right,
Kevin Plaxco.
If you want.
Oh.
Yeah. Sorry.
No.
Me, but that's
okay. Oh, right. So Kevin's here. I hadn't met you before. There you go. Kevin mentioned it. Yeah.
That's right. That's correct.
So Kevin's here he's American from UCSB, but he's been spending a few months of each year in Australia lately, and he's here on sabbatical at MIPS for the moment. And, always up for a chat.
Does Carl want to make a comment, because he's very engaged in that technology?
So I can't say where the technology's at, but I can say they're working on it very seriously.
Yeah.
But what I can say is, everything that's Connie and Rick has described is true. But I think the cool thing about where Kevin's at is he's got feedback control added to it.
Yeah.
Okay. And so we can shape by infusion and bolus the exact AUC you want. Now, the time he spent with me is how we take some of the things that Rika raised around. If you've got an ISF, how do you correlate that back to blood? We're working on that. We're resolving that.
Yeah.
And at an academic level. But, we can, in theory, change the shape of the curve you want depending on your antibacterial.
Yeah, it's super exciting.
I have 2 questions.
Thank you. Is that on? The first point was with what you were saying. Are we going to be able to apply that to continuous infusion vancomycin in hospitals? Because vancomycin dosing has been very problematic, at least at my hospital, even with using a sort of power plan to guide the doctors. So I think that would be really helpful.
I think I missed the first part of your question. I'm sorry.
I was just asking whether any of your work would be able to be applied to continuous dosing of vancomycin.
Right. Yeah. Do we have a comment? Yeah, there have been. Yeah, there have been studies, but not my work per se. Connie, do you have?
Yeah, there have been studies from groups in Sydney, collaboration with Hamburg, that have looked at precision dosing of vancomycin a lot. Yeah. Mm-hmm.
Great.
I use software as well. There's been reasonably good dosing software for vancomycin. That's quite well validated, actually.
Yes, there has been,
Yeah.
and our health service hasn't picked that up yet. But I think Alfred might have been
you can look at the open access software TDMX.
Yeah.
That's pretty good. They have a good validated vancomycin dosing program.
Thank you.
TDMX. I don't have any stake. It's an open access academic software.
Thank you. And my second question was for Dina. I was really interested in what you've done with the bezlutoxumab in the mice with the systemic features. And did you observe any cardiac toxicity in that group of mice that were treated at all?
That were treated or that were untreated?
That were treated with the bezlutoxumab.
We didn't. We didn't look specifically for that. I mean, bezlutoxumab is currently, my understanding is that it's only indicated for recurrent disease. What we've done that's different is that we used it for primary disease, not for recurrent disease. And we did reduce, we didn't see those symptoms of kidney damage, thymus damage, despite the fact that we saw gut damage. We didn't look at the heart. We have looked at the heart in the past with C. difficile infection, and we have seen some signs of damage, but I don't know if those antibodies cause damage.
Yeah, thank you.
Dina, just on that note, has that been looked at in patients? Because, our patients with severe C. diff, they do develop this multi-organ dysfunction, but we always attribute that to hypotension and shock and that they're just generally really sick. But I don't think we've really appreciated whether that might be systemic toxin effects.
Yeah,
and is there something that we could do in that space? Are there sort of measurements or other things that should be done?
There are measures. We have looked at measures of cardiac damage, and they are higher. And I can't remember what they are, Anton, but we have got all of that data in mice, of course, right? Look, from all the work that we've done, I would say that the infection sets in. That's when you see your patients, when the infection has. By that time, you've already got a permeable gut. You've already got lots and lots of bacteria there that are producing a lot of toxin, and that's getting out. The bacteria won't grow anywhere. It's a strict anaerobe, so it's not a problem with the bacteria setting up infection elsewhere. But you have got the toxin being disseminated, and we know from studies on zebrafish that the toxin does target cardiac tissue. So it's a really broad-acting toxin. It has half a dozen receptors that it binds to. And so it does target that tissue. Whether it does that in human tissue, I can't say, but I would say probably yes. It's a really broad-acting toxin. So it's hitting a lot of other tissues. And I think that's an aspect of disease that isn't really appreciated. Yeah.
Thanks. Great talks. I was really encouraged by your presentation, Reka, and your presentation, Connie. In this idea of kind of optimizing treatments, and I was hoping you could both comment from each of your perspectives about some of the logistical challenges of implementing this in the clinic, because I think it is that classic issue of disconnect between the knowledge that we have, but then implementing that knowledge, and particularly from the clinical decision support sort of system as well. Yeah.
Absolutely. And we've talked about that before, and we were even saying, we want to invite opinions from clinicians on the things we are presenting about limitations. So I think, Reka, you had some thoughts on barriers with the TDM part before?
I actually just finished writing a manuscript. We did a study at the Alfred on physician, pharmacists, and scientists, as well as nursing perspectives on beta-lactam and TDM in general, really, and what they thought about it. I think the biggest barriers from a doctor perspective was the fact that they didn't know what to do with the results. They really needed some sort of good dosing advice. The fact that the results were never available enough on time to make a difference to a septic patient. So I think the turnaround times were really important to the physicians. To the pharmacists, it was similar, but they also needed a proper protocol on how to dose these antibiotics. And I think from a nursing perspective, they did not like continuous infusions because it interferes with all their other workflows. So there are a lot of challenges in actually implementing it. And the scientists, of course, they find it quite challenging to have machine time to run the assays, because we all want the results, really quickly, because we want to change the dose in sick patients. So having machine time was a challenge. Having the actual infrastructure and having expertise in the lab is a challenge. And developing the assays and making sure that you validate them and keep them, updated is another challenge from a lab perspective. So we got a lot of perspectives on the challenges to implementation, and we've, I think it's really important to have stakeholders involved from the time. If you decide you want a TDM service and you want new technologies, dosing software introduced, then you need to have your stakeholders informed early, and then you keep them involved throughout the stages of your process. So I've just recently emailed the people who participated in my study saying, hey, I've written the manuscript, I'll send it out to you when it's published. So it's just about keeping that communication going, making sure that they're involved. It is not easy because it's a new test, and it's not easy because there's evidence being generated all the time. So it's basically everyone who's going to use that interface needs to know how they're going to use it, and that they feel that they're always involved and in control of what they're doing for their patients. Yeah.
Yeah, and just a very quick comment. So yeah, like what Rika said at the end about involving the stakeholders. So from my point of view, I would say, yeah, of course we can't do this now, clear. But we sort of, I don't know, overall maybe the community has to decide. We can get all of this data. And like you said, Nenad, you can make lots of predictions from mobile elements. So, but we need to know how to use them, right? And that requires fitting all of the different pieces together. So it's like maybe the community has to decide, is this something we want to do? Or do we want to sort of do what we are doing right now with how it's going? And then if you want to do it, I mean, what I personally would be interested is, I think, have clinicians and working in practice involved in developing these methods to make sure that whatever we come up with, mathematical approaches, translating it to whatever apps may be possible in the future, that actually, it leads to translation, is all I would say to that.
So we're moving on to this we're moving on to having a randomized pilot control trial in the ICU. And part of the project is that we are ensuring that the ICU physicians and ICU pharmacists are involved in dosing, that they already know what the dosing might look like and what TDM looks like. So they have a say in what it should look like. So absolutely involving the people that are involved that will get affected by any changes. So I think that's essential.
Yeah, no, that's great. I think the ICU is an easy bridgehead, given that they're very attuned and they have a captive audience in terms of the patients that they're looking after.
I also think that having it started in one area will help that sort of practice creep in a good way. Yeah.
Okay, excellent.
Yeah, so I'd like to thank again both Rika and Dina and everyone for the great discussion and for the nice session. And then in the interest of time and finishing the day on time, we might go to our coffee break now.
Thank you.